We now move on to one of the most important topics, brain plasticity, the brain's ability to change in response to time, experience, and injury. The brain is not a static structure fixed once and for all. On the contrary, its synaptic networks undergo continuous change. At the level of connections between neurons, we speak of synaptic plasticity, which includes two fundamental mechanisms discussed later in detail. Long-term potentiation, LTP, is a process in which a frequently activated synapse becomes stronger. Signals pass through it more easily, supporting the formation of memory traces. Long-term depression, LTD, works in the opposite direction. Infrequently used synapses weaken, allowing neuronal networks to be cleaned up and organized. Glutamate via AMPA and NMDA receptors is the key neurotransmitter driving these changes. On a broader scale, several types of plasticity can be distinguished. Developmental plasticity is strongest during childhood and adolescence, when the brain is especially sensitive to shaping by stimuli. This is when connections are formed and selected, cortical maps are established, and sensory and motor circuits learn the world. Repair plasticity is activated following injury such as stroke or trauma. Although neurons largely do not regenerate like peripheral tissues, the brain can partially reorganize its connections. Neighboring areas take over lost functions, and new pathways form, enabling at least partial compensation. Experience-dependent plasticity continues throughout life and underlies learning and memory. Every new skill, remembered fact, or habit reflects gradual modification of synaptic strength and number within specific networks. The mechanism of LTP occurring in the hippocampal formation underlies learning and memory. Strong glutamatergic stimulation opens AMPA receptors, leading to depolarization and unblocking of NMDA receptors. Calcium influx activates calcium-dependent kinases, resulting in phosphorylation and an increased number of AMPA receptors in the postsynaptic membrane. The synapse becomes stronger, more sensitive, and capable of transmitting information with weaker stimulation. Let us examine step by step the mechanism of long-term synaptic potentiation, LTP. It begins with strong glutamatergic stimulation. The presynaptic neuron repeatedly and rapidly releases glutamate into the synaptic cleft. This neurotransmitter first activates AMPA receptors on the postsynaptic membrane. Their ion channels open, sodium ions flow into the neuron, and the postsynaptic membrane becomes markedly depolarized. This depolarization is crucial for the next stage. At rest, NMDA receptors are functionally blocked by magnesium ions lodged in the channel. When the membrane is sufficiently depolarized, magnesium is expelled from the pore, allowing the NMDA receptor to open. With glutamate present, the NMDA channel opens and, unlike AMPA, primarily conducts calcium ions. A large amount of calcium 2+ enters the postsynaptic cell, triggering an intracellular signaling cascade. The rise in calcium activates Ca2+ dependent kinases such as KAMK2, as well as other kinases shown on the slide, including PKA and PKC. These enzymes enable the insertion of new AMPA receptors into the postsynaptic membrane and phosphorylate synaptic proteins, including existing AMPA receptors, increasing their conductance. In practice, this means that after the process is complete, the synapse contains more AMPA receptors and they are more efficient. This change persists long after the initial stimulation and ensures that subsequent glutamatergic signals elicit a stronger postsynaptic response. The mechanism of LTD occurs primarily in the hippocampal formation, but also in the cerebral cortex and cerebellum, where it plays a crucial role in fine-tuning neural networks. Long-term synaptic depression allows the brain to selectively weaken less important connections, prevent saturation of LTP, and thus maintain the capacity for further learning. In the hippocampus, LTD contributes to the organization of memory traces and flexible rewriting of spatial representations. In the cortex, it refines sensory and motor maps, and in the cerebellum, it supports motor learning and error correction. Through this process, the brain not only strengthens important pathways, but also actively weakens less useful ones, an essential requirement for selective forgetting, information filtering, and stable operation of entire neuronal networks. The mechanism of LTD, long-term synaptic depression, begins with weak or prolonged low-frequency glutamatergic stimulation. The presynaptic neuron fires less often or at a lower frequency, so the amount of glutamate in the synaptic cleft is smaller than during strong stimulation. Nevertheless, glutamate still activates AMPA and NMDA receptors, but the depolarization of the postsynaptic membrane is moderate, insufficient to trigger a massive influx of calcium. In this situation, NMDA receptors are only partially unblocked and do open, but only a small amount of calcium 2+ enters the cell. It is precisely this dose of calcium that determines the direction of plasticity. A large, rapid rise in calcium 2+ initiates LTP, whereas a small, prolonged increase leads to LTD. This subtle calcium signal does not broadly activate kinases. Instead, it preferentially stimulates calcium-dependent phosphatases, primarily calcineurin and protein phosphatase 1. These enzymes begin to dephosphorylate synaptic proteins, including AMPA receptor subunits and elements of the postsynaptic scaffold. As a result, the properties of AMPA receptors change. They become less conductive, respond less effectively to glutamate, and some are marked for endocytosis, that is, removed from the membrane back into the cell within vesicles. The number of AMPA receptors in the postsynaptic membrane gradually decreases, and those that remain conduct less efficiently. A synapse that previously responded to stimulation with a relatively strong postsynaptic potential now produces a weaker signal. Its efficacy of information transfer declines. In certain circuits, for example in the cerebellum or hippocampus, metabotropic glutamate receptors, MGLURs, also play an additional role in LTD. Through second messenger cascades, they likewise activate mechanisms that remove AMPA receptors from the membrane and reprogram synaptic sensitivity. Let us now pause to consider a phenomenon that reveals the dark side of glutamate, its neurotoxic effects when receptors are excessively activated. This pathological process, known as excitotoxicity, leads to damage or death of nerve cells. It plays a key role in stroke, severe traumatic brain injury, and metabolic disturbances such as profound hypoglycemia or hypoxia. Under ischemic conditions, neurons stop receiving sufficient oxygen and glucose, leading to a halt in oxidative phosphorylation and a rapid drop in ATP production. Without energy, the sodium-potassium pump fails even though it normally maintains proper ion gradients and the resting membrane potential. Sodium begins to accumulate inside cells, water follows osmotically, and the membrane undergoes sustained depolarization. In this state, presynaptic terminals go haywire. They release enormous amounts of glutamate into the synaptic cleft while simultaneously losing the ability to reuptake it, because transporters and astrocytic mechanisms also require ATP. A cascade of excitation develops, involving neighboring neurons and rapidly spreading through the tissue. Excess glutamate uncontrollably activates AMPA and NMDA receptors, leading to a massive influx of ions, especially calcium, into cells. Instead of serving as an excitatory signal, this calcium overload initiates destructive processes. Proteases, phospholipases, and endonucleases are activated, degrading cytoskeletal proteins, cell membranes, and DNA. At the same time, calcium 2+ stimulates enzymes that generate free radicals, resulting in escalating oxidative stress. Mitochondria attempt to buffer the excess calcium but quickly become overloaded. Membrane potential drops, ROS production increases, and eventually the mitochondrial permeability transition pore, MPTP, opens. This causes mitochondrial swelling and complete inhibition of ATP production. In this state, cytochrome C is released from mitochondria, activating caspases and triggering programmed cell death, or, when damage is even more severe, the cell progresses toward necrosis.